Vertebrate adaptive immune system. The adaptive branch of the immune system takes days or a few weeks to become fully active, but it provides a powerful antigen-specific response, it adapts to each infection, and provides a lasting memory of each foreign antigen. If the innate immune system does not kill a pathogen, the adaptive immune system will usually get the job done. The adaptive immune system is made up of different types of T cells, B cells, and APCs, which are all white blood cells that circulate in blood and lymph. More on lymph a little later in the presentation. Each type of white blood cell has a specific job. They communicate with one another and coordinate their responses using cell-to-cell contact and chemical messengers called lymphokines. They strategically remove pathogens, causing as little collateral damage as possible to the vertebrate. The adaptive immune response kills specific targets, unlike the innate defense systems. The key to the specificity is the astronomical number of different antigen receptors on the surfaces of T cells and B cells. Each T and B cell displays a unique receptor ready to bind to an antigen. The antigen receptors of B cells are called antibodies or immunoglobulins. The antigen receptors on T cells are called T cell receptors or TCR. When one of these receptors binds to an antigen, the T or B cell receives a signal and becomes activated. It begins to divide and make more receptors. In this way, a very large number of cells that are antigen specific, that's the key to the adaptive system here, are able to amplify an adaptive immune response. Receptor gene rearrangement, also known as somatic recombination, makes the adaptive immune system diverse and able to recognize an astronomical number of antigens. Early in the differentiation, that is, the maturation of T and B cells, their specialized enzymes make rearrangements in the genes that encode their antigen receptors. Only cells of the immune system can express enzymes that rearrange genes and make controlled mutations. How cool is that? Randomized antigen receptor gene rearrangements in each T and B cell leads to a huge diversity of unique receptors. Now, how does the system adapt to different antigens? Well, there's 3 steps: receptor gene revision or editing, affinity maturation, and clonal selection. Step 1 in antigen adaptation, receptor gene revision. After a T or B cell's antigen receptor is bound by a foreign antigen, the cell begins to divide. Each daughter cell makes additional random mutations in the genes that encode the antigen receptor. We said this a couple slides ago, but this is key, so we've got it here again in bold. Only cells of the immune system can express enzymes that rearrange and mutate specific genes. Some random mutations lead to tighter bonding of the antigen receptor to the pathogen. Cells with improved antigen recognition continue to divide and lead the fight. Second step, affinity maturation. The cells with better antigen receptors undergo additional random mutations in portions of their rearranged genes, generating many cells with different versions of the receptor. Some of these new receptors, by chance, bind even more tightly to the antigen. The cells with those receptors are further activated to fight. Third and final step, clonal selection. The clones of cells with better antigen recognition are activated to lead the fight, and then they divide some more. Their numbers increase relative to poorly responding B and T cells. The white blood cell population matures into one that has higher affinity or binding to the foreign antigen. So, due to receptor gene revision, that was step 1, affinity maturation, step 2, and clonal selection, the quality and effectiveness of the adaptive immune response improve in response to specific antigens. The adaptive immune system has immunological memory, which means the second or third time you are infected with a certain pathogen, you will not get as sick. After an immune response to a particular antigen, a subset of B and T cells will stop dividing, but they will continue to display antigen-specific receptors on their surfaces and can remain in circulation for many years. These cells are memory B cells and memory T cells. They are high-affinity surveillance cells, and they can respond quickly to a similar pathogen before it multiplies. In contrast, the innate immune system does not have a large number of receptors, does not adapt, and does not offer immunological memory. Primary immune responses of the adaptive immune system occur the first time a new antigen is discovered and eliminated. Secondary immune responses occur the next time the antigen appears. Secondary immune responses are faster and stronger than primary immune responses because of immunological memory provided by the adaptive immune system. If mice are injected with an antigen at 2 different times, they will respond much more strongly with antibodies after the second injection. This explains why people sometimes don't get very sick the second time they are infected with the same pathogen and why vaccines work, and more on vaccines later. As we said on the previous slide, the first exposure to antigen A, okay, here's A, and here's the antibody response. Clearly, the higher the better. More antibodies will work quicker and take out the antigen. And here's days after infection. And we have 2 charts here, really. We start time at 0 for the primary response. Then we start it again at 0 prime for your secondary immune response. So let's recap here. The first exposure triggers a primary immune response, which does not start right away. See, we've got a couple days in there before it kicks in. A second exposure triggers the secondary immune response. And that starts pretty much immediately. See that? It's right at 0 days here. And it peaks much higher, much quicker than the primary immune response. And here it is. Many more antibodies and a lot quicker. Primary takes some time before it gets going and does not create as many antibodies. Now, secondary immune responses are specific. They do not improve responses to new antigens. So let's have these mice again. They're injected with a new antigen B at the same time as a previously encountered antigen A. The adaptive immune system will mount a primary immune response to B, because it's the first time they've seen it, and a secondary immune response to A. This pattern makes sense because the immune system has never seen B before, so it mounts a primary immune response. But simultaneously with that, it can mount a specific secondary response to A. The second exposure to antigen A can occur in the same time frame as the primary response to antigen B. But since it's the secondary response, it happens quicker and is more effective. So let's look at our chart. We'll start with antigen A. And here's the first response. Not that very high in antibodies, and it doesn't start right away. There's a time delay there. Second time around, starts immediately, reaches a very high amount of antibodies, and then comes off as they've wiped out the pathogen there. Let's look at what happens to B. B, it's the first time the system has seen it. So it starts here, and it starts a few days later, and is certainly not as strong as the secondary response to antigen A. So what have we seen in these 2 systems? Well, the adaptive immune response takes days or weeks to develop, while the innate immune system prevents infection or works immediately with seconds, minutes, or hours. The adaptive immune system can respond to any antigen, while the innate immune system recognizes only common structures on pathogens. However, the good thing for us is the 2 branches work together. If a pathogen is not completely removed by the innate branch then helps to activate. It's kind of like it's calling for help from the adaptive branch. The adaptive immune response, it actually gets stronger during an infection because the selected B and T cells adapt their receptors. In contrast, the innate immune response has 1 level of response for each pathogen. An adaptive immune response leads to memory of the pathogens because it leads to the development of memory B and memory T cells. In contrast, the innate immune response does not have memory cells. Let's work an example where we talk about the cooperation between the 2 systems. We're going to start with the mild COVID-19 infection caused by the SARS-CoV-2 virus. If the initial exposure to the virus is low, It does not overwhelm the innate response. The viral load in the bloodstream will not get as high, and the adaptive response removes the virus after a short period of symptoms. It doesn't have as much to do. So here's your innate response. Here's the viral load. Starts off, doesn't get terribly high. And then it's already coming down by the time the adaptive response kicks in. So the symptoms don't last very long. But if the initial exposure is high and the virus proliferates early, and here's your viral load increasing very quickly, and it gets past the innate response, right? The innate response is doing its best, but it can't manage it totally. The symptoms will last longer and become more intense before the adaptive immune system, see, it's starting here where it's already peaked. Okay, so the adaptive response takes a little time to get going, and it takes more time for it to get the infection under control.